I guess I will go ahead and start with my introduction of Yusan. So it is my great pleasure to introduce Yusan on the day of her defense of her PhD. Yusan came to my lab back in 2014 when my lab was still at Tulane University, and she came with a bachelor's degree from National Taiwan University and a year of experience working as a research assistant on an animal behavior project at National Taiwan Normal University. The focus of my lab's research on the super cool frog that you're going to hear about today has, like most sexual selection research, been focused on the female side of the equation. But Yusan quickly convinced me that there was merit in figuring out the male side of the story, the fight over access to females. So studying animal behavior is hard work for those of you who haven't done it before, especially when it means sitting motionless in a tropical forest, getting eaten alive by bugs for hours on end, and waiting for frogs to finish courting and get on with it already, or waiting for cross-fostered offspring to reach the age of sexual maturity, which for these frogs is about a year, so that you can ask them who they prefer in a mate or they prefer to fight with. But Yusan is patient, and she's hardworking and clever, and all of this has paid off in the dissertation you're about to see. She published the first results of her dissertation in 3 of the top journals in our field of evolutionary biology and behavior. She published them in Evolution, the Journal of Evolutionary Biology, and in American Naturalist. Then she followed that up with a first author publication in one of the top journals in all of science. She had a publication that made the cover of Nature. Over the past year, she's won awards for both her research and her teaching. She earned an award in the Society for Integrative and Comparative Biology's Best Student Paper competition in January, and she won our department's Stanton C. Crawford Graduate Student Teaching Award this past year. So while I haven't seen her teaching in the classroom, I've heard great things, and I'll share a quick anecdote that I think illustrates her teaching skills from the time that she spent last summer at the Pima Tuning Lab as a TA. So for those of you who don't know Yusan as well as I do, Yusan loves volleyball. And when she came to PLE, the Pima Tuning Lab, she saw immediately the sand volleyball court that we had. And she saw the 50 or so undergraduates that were around and thought, okay, here's my volleyball team. Unfortunately, and I can tell you this from experience, the undergraduates were not very good at volleyball. Chris, my assistant director, and I had tried playing volleyball with the undergraduates, and I think over about an hour of playing volleyball, we got maybe 1 or 2 volleys across the net. It was pretty sad. So during the time that Yusan was TAing, I heard a lot of volleyball going on, and I'd seen a lot of volleyball playing. And then one day I walked by the court and realized that there was an actual volleyball game going on with very long volleys, good points, skill being shown by the students. And I realized that Yusan had coached these group of undergrads into her volleyball teammates, and she had gotten them to play every day while she was there. It was really quite an impressive change. So as Yusan has prepared to finish her PhD, she took time out from her writing, her volleyball, and her research to write some proposals in application to some of the most prestigious postdoctoral fellowship programs around. She was rewarded with offers from 2 of them, and had to turn down an offer from the University of California's President's Postdoctoral Fellowship Program in favor of her top choice, a fellowship from the Living Earth Collaborative at Washington University in St. Louis. So she'll be headed there to begin an exciting project that she designed on Trinidadian guppies later this summer. Yusan has been such a pleasure to mentor and has been such a valued member of our lab group. It's hard to imagine what the lab would look like without her. Well, at this point, it's actually hard to imagine what the lab looks like at all, but we're definitely going to miss Yusan's face around the lab. But I know it's time to let a student go when you get to the point where you're pleased with yourself when you have anything you can criticize in her dissertation, and you start to wonder who's advising who, and that's where it is between me and Yusan. So as I wrap up here, I'll just say that I couldn't be prouder of all that Yusan has accomplished, and I hope that from here on out, she'll continue to see me as an enthusiastic collaborator and a cheerleader, but no longer an advisor. So now I'll let Yusan take over the screen and present her dissertation, which is entitled Divergent Mating Behaviors and the Evolution of Reproductive Isolation.
Okay, thanks, Corey. Corey, have you muted everyone in the
I will mute
chat?
everyone.
Okay, thanks. All right. All right, I need to first unmute myself. Sorry about that. Share. Okay, now we can get started. So thanks so much, Corey, for the awesome introduction. It has always, I have always been very grateful to be a part of your research group. And so I'll get started. So thank you everyone for getting up bright and early to come to my talk. So this is actually not how I imagined I would defend my dissertation, but here we are, and I hope you enjoy the talk. All right, so sexual selection, as we know, is a very strong force of evolution, and it has created some of the most bizarre and extreme forms of function that we see in nature. So these are just some of the examples that we have that are more visual, because we humans are visual animals, but in nature, this can happen in all kinds of different modalities. And so in addition, sexual selection is believed to be a very strong driver of speciation as well. So imagine if we have a population with two different phenotypes, and if the mate preferences, so on how the females select their mates, evolved along with these divergent of sexual traits, what we will get is a mating pattern called assortative mating. And so this basically means that individuals will tend to mate with individuals that are phenotypically similar to their own. So this mating pattern can potentially reduce gene flow among these two divergent phenotypes and affect behavioral isolation between the two groups and potentially lead to speciation. So this represents the core mechanism of speciation by sexual selection, and is pretty much how our Bio 101 textbook teaches us about the process. However, there are a couple of non-trivial aspects of sexual selection that I think is missing from this traditional paradigm. So for one, as Corey has just mentioned, I'm really interested in the other side of sexual selection that is missing from this view. So in a system, when you have females choosing amongst the males, then at the same time, you will also have a number of males that are competing with each other to access these limited number of females. And so the thing is that these divergent traits, so the coloration that I was just talking about here, these key sexual traits oftentimes not only mediates in female choice, but oftentimes also functions in determining the patterns of male-male competition. However, we have comparatively a lot less understanding in how these male-male competition patterns influence speciation. So the second thing I want to talk about is the development of animal behavior. So dating all the way back to the era of ethology, when under Divergent's framework, we have long known that animal behaviors can be mediated by a couple of different processes. So animals can behave certain ways because they have the genes that coded them to do so. Or alternatively, animals can behave certain ways when they have some past experiences that will influence their current decision and influence their current behavior. And then so despite knowing that both genetic mechanisms and learning may be important in the development of animal behavior, traditional speciation research have focused on genetic mate preferences. So that builds up pretty nicely to the framework that I'm going to talk about today, and also my full dissertation framework. So in short, I hope to bring these two important yet understudied processes in sexual selection into the story of our understanding in speciation by sexual selection. So for one, I wanted to know that in addition to female choice, how does male-male competition influence the evolution of reproductive isolation? And secondly, how does adding the layer of behavioral learning contribute to our understanding of how this process works? So I have been using a lot of fish figures to talk about my concept, but today I'm actually going to talk about frog behaviors. So this is my study system. So these are the awesome strawberry poison dart frog, Uphaga familial, and they are a tiny species, about 1 inch long or 2 centimeters long, that distributed in the tropical forests in Central America. So their distribution across Nicaragua, Costa Rica, and Panama. And throughout most of the distribution range, they have this very iconic coloration of red body and blue limbs. However, if we zoom into this area called Bocas del Toro Archipelago, what we will actually see is that on these different islands and also adjacent mainland area, we have this amazing variety of different color morphs. So these different color morphs, they diverged in less than 10,000 years. And also these different color morphs are capable of interbreeding with each other and producing viable and equally fertile offspring. So they're still considered one biological species, just at the beginning of a divergence. And so in addition to this very interesting fact, they also have a really cool life history. So in the species, the males compete for territories. So they will try to wrestle each other and compete for dominance. And the territory is very important to them because it is where they vocalize and try to attract females. So the females that pass by the male's territory, if she is satisfied with the male's courtship, the pair will then mate and lay a clutch of eggs in the male's territory. So these eggs, they are terrestrial eggs. So the highest mortality of them is from drying out or desiccation. And so the main parental role of these males is to pee on the eggs and keep them moist. So awesome parenting skills there. After the tadpoles are hatched, the females will then come back and transport the tadpoles on their back into water bodies. So for example, water is trapped between plant leaves like bromeliads, and she will feed them with unfertilized eggs until they metamorphose. So this is a system with very intense male competition, strong female choice, and also parental care of both sexes. So people have been taking advantage of this amazing system and asking the fundamental question of speciation by sexual selection. So namely, are the female mate preferences evolution tracking the evolution of coloration? And so indeed, in these studies, including some of Corey's work in the past 2 decades or so, people have demonstrated that yes, among most of the color morphs being tested, these females seem to display what we call associative preference. So in other words, if you offer a female a choice between a male that has the same color and a male that has a different color, then oftentimes the female will choose to associate with a male that has the same color. So these patterns, they have been interpreted as reproductive isolation evolving among these different color morphs. But as you might have noticed, this interpretation did not take into account how male-male competition is operating in the system, and also haven't considered how these color biases came about. So whether they are genetic or they are learned. So that builds up pretty nicely for my dissertation. So we have a system that we know quite a lot about, but is also missing these 2 factors that I'm really interested in. So in today's talk, I'm going to break the content into 3 big aims. So first, what we did is we use a more kind of observational approach and looked at whether male competition, in addition to female choice, is mediated by coloration, so the divergent sexual trait in these poison frogs. And secondly, upon establishing how male-male competition pattern and female choice pattern is influenced by color, we then set out to experimentally test how these 2 processes in sexual selection interact with each other. And lastly, we tackled the question of how these color biases came about, so whether they are genetic or whether they are learned, and also upon knowing their proximate mechanism, how does that change our understanding of how reproductive isolation evolved? All right, so jumping directly into the first aim. So we took advantage of an area also in the Bocas del Toro that is a red and blue phenotypic contact zone. So in this area, we have populations that are entirely red and some populations that are entirely blue. And in between these two areas is an area called Dolphin Bay, where we can find both a red morph and a blue morph, and in addition, some individuals that seems to be phenotypically intermediate between red and blue, and also in different frequencies as well across this region. So this presents a very unique opportunity for us to not only look at whether behaviors have evolved along with the coloration, but also whether behavior patterns are the same when the color morphs are in allopatry, so when they are in their own populations, compared to when they are in sympatry, so when they are different color morphs are in the same location. So the first thing we tackle is the basis of traditional speciation bisexual selection, so female preference. What we did is that we went out to select 3 different populations. So we have our entirely red population, our entirely blue population, and a polymorphic population where we collected all 3 color morphs. So what we did is we brought these females back to the research station at Bocas del Toro, so the Smithsonian Tropical Research Institute, and then we assayed the females for their preference. So we offered the female over here a choice between a red male, a blue male, and a phenotypic intermediate male. So this is on a test arena view from above. So on each of these 3 males are confined under transparent plastic domes, and the female is free to roam around the arena. So we use the time that the female spent in proximity with each of these 3 males as a proxy of a preference, which is going to be shown here in the y-axis. Okay, so the pattern that we found first is that in the entirely red population, the females seem to prefer the red male over the other 2 options, whereas in the blue monomorphic population, what we saw is that the females prefer the blue color over the other 2 options. So it seems like so far we are following the patterns that people have found in other color morphs. So basically the evolution of female preference is closely tracking the evolution of coloration. But what I think is very interesting is when we started to look at the behavioral patterns in this polymorphic population, what we saw was actually this. So instead of seeing the red preferring red, blue preferring blue pattern that we might have expected from the allopathic population, what we actually saw is that regardless of the female's own coloration, everyone seems to prefer red over the other color morphs. So this suggests that the divergent preferences that we observed in allopatry does not necessarily predict what is happening in the sympatric population. And then so we need to be really careful when we are interpreting divergent behaviors that happen in allopatry, because things might still change in sympatry. So this is the female side of things. We also look at the pattern of male-male competition in relation to color. So did a kind of parallel experiment or investigation across the same red and blue phenotypic contact zone. So this time we're using a slightly different method. So for females, we actually require to use actual males because they're really hard to trick. But for the males, we can actually use fake frogs and do it in a more controlled setting. And so these are 3D printed model frogs, and we painted them to match the actual coloration of the frogs. And so what we did is we put a servo control attached to this frog so we can jiggle them around. And we have a Bluetooth speaker where we can play on the call playbacks of aggressive calls, and then a video recorder to record everything. So a little bit of fun backstory about this robot frog. So at that time when I was designing this robot frog, I was looking around for a good container to put all my wirings and batteries in. And then I came across this awesome product on Amazon when I was not even searching for the container. So this is a planter that's shaped like a log, and it provided the perfect solution because it's a planter, it's waterproof, it's durable. So I have been using the same planter for 2 field seasons, and it's still really good. So 5 out of 5 would recommend. So anyways, what we did is we brought our blue models and our red models across that phenotypic transition zone and assayed the males for their aggression. So what we did is called a simulated intrusion experiment. So the first thing that we did is look for a male that is calling, which means that he is territorial. And then we will put the whole apparatus into his territory. And then after a couple of minutes of acclimation, we will start jiggling the frog and start to play the call playbacks. And we will record the behaviors that the territorial male respond towards this simulated intrusion. So this is what it looks like when we actually did the experiment. So that's our model frog and our really angry territorial male. So this experiment was actually pretty fun to do, despite all the mosquitoes. So anyways, we did it across the transition zone and also with different color stimulus models, and also did it with different frog color morphs. So this is what the pattern looked like 372 trials later. And so we have this time 4 populations. So we have our entirely red population, our entirely blue population, and 2 populations that are polymorphic. And so one of them has a higher red proportion, and one of them has a higher blue proportion. So because we were interested in frequency-dependent effects at the time. And so y-axis here is proportion males attacking. So this basically shows that out of all my trials, what percentage of the males end up escalating the defense to the highest aggression level, which is attacking the model, as you have just seen in the video. And the response to red and blue will be shown in the respective color bars respectively. All right, so the pattern we saw was that in the entirely red population, we saw that the males are a lot more aggressive when the intruding model is red. And in the blue monomorphic population, what we saw instead is that the males are more aggressive when the intruding model is blue. And so it seems like so far we are still following the pattern that is similar to what we saw in the females. So basically, the individuals are more responsive when they are encountered with a color that is similar to their own, or you can say similar to the color of the population that they are in. So again, when we look at the 2 polymorphic population, we again see a very different pattern from what we expected to see. So what we saw is actually, regardless of the male's own coloration and regardless of the intruding model's coloration, everyone seems to have about equal amount of aggression towards the intruder. So there are some slight patterns within these, but nothing as significant or as clear of a pattern as the 2 monomorphic population. So it seems like, again, we're seeing this divergent behaviors in allopatry, but then it breaks down again when we have different color morphs in the same population. Okay. So a short summary summing up this part of the 2 experiments. So first, by using these behavioral assay, we have clearly demonstrated that indeed both female choice and normal competition in our poison frog is mediated by coloration. So this is the dual utility of sexual traits that we were talking about. And secondly, via comparing the behaviors in allopatric populations versus sympatric populations, we saw that divergent behaviors in allopatry doesn't necessarily mean that there are behaviors that will be continued in sympatry, because things might change in sympatric populations. And lastly, we talk a little bit about what could have caused this discrepancy between allopatric populations versus sympatric populations. So there are a variety of different possibilities. So including it could be introgression of the behavioral genes if these behaviors are genetic. So these mating between different color morphs could contribute to behavior changes. Or it could be because selection is different in sympatric population versus allopatric population. Or it could be because that these behaviors can change with the social environment, and the social environment being in a population with multiple color morphs and just one color morph is simply different. So by this point, it is really hard to distinguish which one of these is the real hypotheses or the correct hypotheses, because we don't know yet what is actually contributing to shaping these behaviors. So this is something that we will come back to in the later part of my talk. But upon following this part, what I first want to talk about is how these 2 processes, so now that we shown that both of them are influenced by color, how these 2 processes are interacting together to determine the mating patterns. So going back a little bit to the concept of the utility of sexual traits. So from the previous 2 sections, the two chapters, we have established that the coloration is indeed mediating both female mate choice and male competition in our poison frogs. So in a couple of other systems that also have this dual utility of sexual traits, one pattern that people have found over and over again is that these males that are of different phenotypes, usually they are not symmetric in their competitive probabilities. So some phenotypes will be a lot more aggressive than the others, for example, and that could contribute to dominance asymmetry between these two phenotypes. And so this is actually something that is seen in our poison frogs as well. So this was a study looking at the co-evolution between personality, so boldness and aggressiveness, and coloration. And so overall, what they found is that some morphs are a lot more aggressive than others. So this potentially could contribute to competitive asymmetry if these different color morphs were to interact with each other. So what that means for the evolution of reproductive isolation is that these male-male competition, which oftentimes happens before female choice, will disproportionately eliminate some males from the mating pools in which the females can choose from. So in other words, even if a female possesses a preference for a certain trait, if that male is not capable of making it to the point where the female can select them, then this preference might not be able to be expressed at all. So in this specific section, I will be testing for the hypotheses that male-male competition, or the results of male-male competition, can limit the female from expressing her preference and choosing her preferred phenotype. So to test this hypothesis, we designed an experiment with two treatment groups named attractive winner and attractive loser. So in the attractive winner group, we have three individuals in a tank. So we are offering the female a choice between a winner male, so the territorial male that also has an attractive color, and a loser male that has a relatively unattractive color. So in this treatment group, we have male-male competition and female preference operating in the same direction. So whereas in the other treatment group, we are offering the female a choice between a territorial male that has an unattractive color and a loser male that has the attractive color. So in this case, we have male-male competition and female preference operating in opposing directions. So by comparing the mating patterns in these two treatment groups, we can then kind of tease apart the relative importance of male-male competition and female preference and any potential interaction between them to determine the mating patterns. So this was an experiment done at University of Pittsburgh in a breeding colony of the poison frogs in the Richard Zawacki lab. So in the colony, we have a couple of different color morphs, and we have selected three to do this experiment. And so these three color morphs, they have also been demonstrated before to show a sort of preference. So basically, the females prefer to associate with males of the same color. So this is how we set up the experiment. So this is a typical breeding tank in our colony. So in there we have a plant where the females can lay eggs on, and 4 PVC pipes with water in them. So this is mimicking bromeliads in the wild. So where the females can drop their tadpoles in after they hatch. So what we did is we introduced 2 males of the same size but of different color morphs and let them compete for dominance. So this is what will happen when you put 2 males in a confined space. Basically, they fight. So it's really fun to watch them fight. I always like to compare them to 2 gummy bears trying to squish each other because they don't have claws or teeth to hurt each other. And kind of as a consequence, this process can go on for very long. So it can take hours or sometimes even days. I've seen one that have taken on and off on like 3 days when I was observing them. And so what we did in the colony is that we constantly go back and check in with the frogs and see how they're doing until we have a clear winner and loser in the tank, which is defined by their behavior. So upon having a clear winner and loser, what we did next is to introduce the female. So in the tanks designated to attractive winner, we will introduce a female that prefers a winner's color. Whereas in the other treatment group, the attractive loser group, we introduced a female preferring the loser's color instead. So what we did is we then keep these trios together until they produce tadpoles and put them in one of these PVC pipes. We then collected the tissue samples from these 3 individuals and also all the tadpoles and used microsatellite markers to genotype them and figure out the paternity of these tadpoles. So this will tell us directly what the mating pattern is, and also the relative reproductive success of these two different males in the two different treatments. All right, so in this slide, I'm going to show you the results from the mating patterns. It looks a little bit complicated, so I'm gonna break it down for you. So first, just to remind you, we have two treatment groups. The female is either offered a choice between an attractive winner and an unattractive loser, or she is offered a choice between an attractive loser and an unattractive winner. And on the right, I will be showing you the mating patterns. So on the x-axis, we have our 4 color morph combinations. So the photo and the first color is the color morph of the females, and the 2 colors in parentheses are the 2 options that the females were given in this choice trial. And a y-axis is proportion of the trials in which the females mated assortatively. So in other words, this is a measure of the degree to which the females mated according to their preference. So whether they follow their preference or they mated with the other male. And the 2 treatments will be shown in darker and lighter shades respectively. So just to set up some of our prediction what the data should look like. So in a null hypothesis, if mating is not influenced by female choice or a male competition, what we should see is around 50% mating chance across the board. So basically mating is random. Alternatively, if female color preference is the stronger driver of mating patterns, what we should see is that the females should primarily mate according to her preference. So everything should be above the 50% line, and we won't see a clear difference between the territorial treatment groups. Alternatively, if male territorial status is the main driver of mating patterns, what we should see is a clear effect of territoriality treatment. So basically, the females should mate with an attractive male when he is the winner, but not when he is the loser. Okay, so with these predictions in mind, this is the pattern that we saw. So this is from 88 different trials that are distributed in the different treatment groups and the different color morph combinations. So overall, although there are some differences among the different color morph combinations, overall we see a very strong effect of territorial treatment, and the direction of these bars are exactly how we predicted under the second hypothesis. So this supports our hypotheses that male competition does indeed in these poison frogs limit the female from expressing their color preferences, which I think is pretty cool. And so in addition to the main results that we found, there is also a pattern that we saw in this experiment that I think is worth mentioning. So in most of our setup, the loser male in these tanks usually don't do much. So usually they don't engage in courtship with the females, and they also don't attempt to fight with the winner males again. However, there are 34% of our tanks, the loser actually started to get antsy and started to try to fight the winner male or try to court with the females after the females were introduced. And so we're interested in whether this behavior contribute to mating pattern differences, or does that change the reproductive success of the losers and the winners. So we separate these 2 categories and look at their reproductive timing. So what I'm going to show you is the reproductive timing, which is going to be an accumulation curve on the loser's behavior. So depending on whether they're submissive or aggressive in our tanks. And this is what we found. So very surprisingly, these aggressive behaviors or aggressive loser are actually not more reproductively successful in getting matings. So we see approximately the same reproductive timing and approximately the same reproductive success by the end of the experiment, which is cut off at 200 days. However, when we look at instead on the reproductive timing of the winners, so the other male in the tanks, what we saw is a very significant trend. So what we saw is that in the tanks where the losers are aggressive, these winners, these territorial males, are actually reproducing a lot more slowly. And also by the end of the 200 days cutoff point, less than half of them successfully reproduce. So it seems like these aggressive behaviors displayed by the losers, they are not increasing the reproductive success of the loser themselves, but they are inflicting costs on the other male, so the territorial male in our tanks. Okay, so to sum up on this breeding experiment, what we demonstrated through this control breeding experiment is that male-male competition, as we hypothesize, does indeed limit the female from expressing their color preference and choosing their preferred color. So this has potential impacts on reproductive isolation because it will limit the female from translating their assortative preference pattern into assortative mating patterns. And secondly, from that reproductive timing analyses, we have shown that males who receive more aggression or more interference during courtship have lower reproductive success. And this holds true even when they remain the territorial winner. So this thing will become, this result will become important later on. So, I will come back to this point a little bit later. And lastly, although it should be clear by this point, I just want to re-emphasize that mating preferences does not equal mating pattern. And so when we want to look at mating pattern, which is the most important thing in reproductive isolation, we need to look at both female preference and male-male competition. So that concludes the second part of my talk. And lastly, I will use the remaining time to tackle the question of how these behavior biases came about. So how does females develop their mating preferences? How does the males develop their aggression biases? And how these proximate mechanisms can change our understanding of how speciation by sexual selection works. Okay, so as I mentioned in the beginning, mating behaviors in traditional speciation models are oftentimes considered genetic. However, there are also a lot of mating behaviors that can be shaped through learning, so perhaps more common than we thought. And this can create very different evolutionary trajectories compared to genetic behaviors. So one of the learning modes that have caught the attention of evolutionary biologists is something called sexual imprinting. So sexual imprinting is a form of learning that describes when females learn parental traits and use them as a model for species recognition after they're sexually mature. So the most famous example is perhaps Lorenz's Gosling experiment. So when these baby geese, if the first thing out of their shell that they see is a human, then they will recognize human as their own species, and they will follow the human around during their Gosling stage. And when they're sexually mature, they will attempt to socialize with human and also attempt to display courtship behaviors to humans as well. And so this type of learning is very interesting to evolutionary biologists because it is a method that it is a mechanism that can generate co-divergence between trait and preferences. And so if you imagine in a population, you have a novel trait or a new trait, that mating trait that evolved, then through imprinting, the offspring of that novel individual will automatically develop a preference for that trait. So in this case, we don't need two independent evolution events for trait and preference to evolve together. Just through this one evolution and imprinting, we can have both. So this is what we termed, if you're familiar with it, the one allele mechanism. And so this is a learning pattern that has been found in a lot of birds, so more than 100 species of birds, in some mammals, and more recently demonstrated in some fishes like cichlids. So you might notice that amphibian is not up there, and that is because by this point there isn't any evidence on imprinting in amphibians. And so we've looked at our frogs, and as I said earlier, these frogs has a very elaborate parental care. So the tadpoles rely entirely on the mom's fertilized eggs as their only food source. And so when a mom visits their tadpole, the tadpole will actually exhibit this vibration behavior to beg for food. So a lot like a baby bird begging their mother bird for food. And so we hypothesize that during this process, the tadpole can learn the mother's coloration and later use this as a model to develop their mating preferences if they're a female, or their aggression biases if they are a male. So we designed an experiment to test this hypothesis. So this is also a controlled breeding experiment or a rearing experiment that has 3 different treatment groups and also done in the breeding colony at the University of Pittsburgh. And so we have 3 treatment groups. So in a purebred group, we have an offspring produced by parents of the same color. So this is our control group, business as usual. And then we have our crossbred group in which the offspring is produced by 2 parents of different color morphs. And then lastly, in a cross-foster group, we swap the tadpoles between 2 differently colored parents and let them raise the tadpoles, feeding them unfertilized eggs until they metamorphose. And so after metamorphosis, we kept these juveniles in their isolated tanks until they are sexually mature, and then we assay them for their behavior. So for the females, we assay them for their preferences, and for the males, we assay them for their aggression bias. And so in the purebred group, we assay the individuals for their association toward their own color or a contrasting color. In the crossbred group, we ask them to choose between the mother's color and the father's color. And lastly, in the cross-foster group, we ask them to choose between the foster parent's color and the biological parent's color. Okay, so and by comparing the behavioral pattern across these three treatment groups, we can then tease apart these two hypotheses. So basically distinguishing whether these color biases are genetically inherited or whether they are shaped by learning. So in the slide, I'm going to show you the results from this experiment. So we have female preferences on the left and male aggression biases on the right. And on the x-axis, we have our three treatment groups. So our purebred group, our crossbred group, and our cross-foster group. On the y-axis, we have association type proportion. So this is basically on an indicator of the behavioral biases of these individuals. So anything that deviates from this 0.5 line means that there is a significant preference, so in the female preference case. And the identity of, or the direction of their biases are labeled on the top and the bottom of each graph. So what we saw in this experiment is first in the purebred treatment, what we saw is that these offspring grew up to prefer their own color, or you can say their parents' color, over a contrasting color. So this is great because that basically means captive breeding is not interfering with the development of mate preferences. And so in a crossbred group, what we saw is very interestingly that these offspring grew up to prefer the mother's color compared to the father's color. So this basically means that whatever influence they're getting from the mom is more significant than that of the dad. And lastly, and I think most excitingly, is in this cross-foster group. So what we saw is that these offspring grew up to prefer the foster parent's color compared to the biological parent's color. So this is very exciting because this is a really clear demonstration that these behaviors are not developed through genetic inheritance, but instead they are shaped by learning, and more specifically on learning or imprinting on their mother during the tadpole stage. So we then look at what happens with the male offsprings. So we see pretty much the same pattern. So in a purebred group, they prefer their own color. In a crossbred group, they prefer the mother's color. In a cross-foster group, they prefer the foster parent's color. So overall, it seems like in our poison frogs, the behaviors of both males and females are shaped through learning, and more specifically on imprinting on the mother when they are a tadpole. And so I think this is really exciting because, as I said earlier, this is the first evidence, as far as we know, that demonstrated imprinting in the amphibian. So this opens up a whole new lot of questions on how other amphibians with parental care, whether they also have imprinting, and also for those species that doesn't have parental care, is any form of learning during the tadpole stage going to influence their mating behaviors later on? So I think there is a lot of possibility of this research avenue. Okay, so in addition to the excitement of figuring out what is behind the development of these behaviors, this finding is also very significant in the evolution of reproductive isolation. So as I briefly mentioned earlier, genetic behaviors and learned behaviors can have very different evolutionary outcomes. And so, so the traditional models, as I introduced, mostly consider preferences to be genetic. And there are also some more recent works on the role of learning on speciation that looked at how imprinted mate preferences can influence the speciation process. However, out there isn't a model yet at this point that looked at both learned male competition and learned female preference at the same time. So this is what we set out to do in this last part. So we developed a population genetics model to explore the evolutionary implications of these imprinted behaviors that we found in the poison frogs. So to talk about the model, we need to take a step back and look at some of the mathematical foundations of traditional speciation by sexual selection. So these group of models that assumed the mating traits and the preferences are both genetically inherited is now what we loosely term the Fisher-Landé-Kirkpatrick model. And so briefly, this model, how it works is that divergent female preferences can segregate the phenotypic variance in a population into two reproductively isolated groups. And so mathematically, this is a plausible scenario, but one of the consensus of these FLK models is that this is a very difficult process to occur, and it requires really strict conditions. And the reason behind that is because there are two main hindrances that this process needs to overcome in order for speciation to happen. So the first one is the difficulty of maintaining mating trait variation during divergence. So that is because selection via female preference is something what we call positive frequency dependent. So you can think of this as a selective pattern that if you have more of certain phenotypes in the population, you will have even higher fitness for that phenotype. So this whole thing will kind of like snowballs down or run away and very quickly turn from your original divergent selection and change the system to a directional selection, and you lose one of your mating traits. And so obviously in this case, you can't proceed to speciation because you don't have the other divergent evolution lineage. So that's the first challenge. And the second challenge that these models have identified is the difficulty of maintaining the association between the trait and the preference. So remember, these traditional models assumed both the mating trait and the preferences to be genetic. And so it's very difficult to maintain two alleles on two different loci when there are a tiny bit of hybridization going on with these two lineages. So when that happens, it will again snowballs down, and what you will see is a mismatch between the mating traits and the preferences for those traits. So by this point, you basically get pretty much close to random mating, and that will break down your putative reproductive isolation, and hence you don't get speciation. So with these two challenges, again, a lot of additional strict conditions for speciation by sexual selection to happen. So people have been doing things like adding additional divergent natural selection to stabilize on the mating trait variation, or if the loci of the trait and the preference is very tightly linked physically, then that will also help with speciation. So inspired by what we found in poison frogs and also some pioneering work on how learning drives speciation, we hypothesized that by incorporating the two factors that I was talking about, so parental imprinting and male-male competition into the speciation models, we can overcome these two hindrances. And so we built a population genetics model to test this hypothesis. So this is a diploid population genetics model, and it's a deterministic model. And so I will walk you briefly through the build-up of this model. So basically in a population, we have males and females, which each of them has a coloration or a mating trait that is determined by a single locus that has two alleles. And also these individuals also have their behavioral biases. So the males have their aggression biases, and the females have their mating preferences, which will be indicated by the color filling in the circles. So you might notice that there are some mismatches of coloration and behavior, and that is because if you imprint only on one parent, there are some chances that you will get this mismatch in your offspring. We'll talk about that a little bit later. But the life cycle of this model includes 3 steps. So in the male-male competition step, the males, we use the aggression biases of the males to calculate the relative amount of aggression that each color morph receives. So remember, back in the second part, we have demonstrated that males who receive more aggression are less likely to reproduce. So we can use this information and calculate the effective frequency of the male color morphs that enter the mating pool in which the females can choose from. And so in the next step, female choice, because female have their mating preferences, they will choose the males based on these preferences and also encounter probability. And so in our setup, the females follow strict polygyny, which basically means that all the females have equal reproductive success, and direct sexual selection is only operating on the males. And then so using this pairing information, we can then move on to the reproductive imprinting stage and predict the color morph distribution and also the behavioral bias distribution in the next generation. And then so we then can come back again to our male-male competition and set up our recurrent equations this way. And so this is the setup of our population genetics model. And so the way that we analyze these models is that we focused on two criteria that are related to the two challenges that I just talked about. So roughly speaking, a polymorphic population has three potential evolutionary outcomes. So first, it might lose one of the mating traits and goes to fixation. So obviously, at this point, you don't get speciation. And even if you maintained your mating trait polymorphism, you still need a tight association between your trait and your preference in order for reproductive isolation to develop between these two groups. And so first, looking at the scenarios that can lead to mating trait variation maintenance, this is a contour plot looking at two of the parameters that we can toggle in our population genetics model. So on the x-axis, we have female preference strength. So you can think of this as on how strong the selection via female choices. And on the y-axis, we have male aggression biases on beta. So you can think of this as the selection via male-male competition. So what we did is we look at different parameter combinations and figure out whether the condition can successfully maintain the polymorphism in a population, or it could lead to fixation of the system. And if we did that over on all the possible combinations, this is what we saw. So breaking down the pattern a little bit, what we can see here is that as we increase selection via female choice, what we saw is that it is less likely for the system to maintain a stable polymorphism. And this is because, as very similar to the genetic female preferences, the selection via female preference is positive frequency dependent and will tend to pull the system towards fixation. Whereas if we look at the y-axis, as we go up the y-axis, it is more likely for the system to maintain stable polymorphism. And this is because male-male competition, in this case, will generate negative frequency dependent selection, and that will tend to stabilize the polymorphism in the population. Okay, and then so after getting our stable polymorphism, we still need to figure out how we can get this tight trait preference association and what scenario might contribute to the highest trait preference association. So what we did is we calculate something called the phenotypic linkage disequilibrium. So if you're familiar with linkage disequilibrium between two different loci, this is very similar, just that it's between a genetic trait that is diploid and also a phenotypic behavior, which is essentially haploid. So what we saw in this part is that the association between the trait and the preference is actually not associated with the male-male competition strength, so beta. However, it increases with female preference strength alpha. So as we have stronger female mate choice, we have a higher association between the trait and the preference, and also a higher probability that the system could proceed to speciation. So taken together, reproductive isolation or speciation is most likely to occur when we have high female preference, because that will lead to high association between trait and preference. But also we, at the same time, need strong male-male competition in order to sustain the high alpha that we have here to maintain the polymorphism in a population. So breaking it down mechanistically, the implication of these imprinted behaviors is that rival imprinting, which is the imprinting that generates aggression biases, this is the key to stabilize the polymorphism to maintain both mating traits around in a population. Whereas sector imprinting, so imprinting that generates mate preferences, are the key to maintain this trait preference association that will later potentially lead to reproductive isolation among the two different phenotypes. So short summary from this relatively long part. So first, through the rearing experiments, we have demonstrated that both male behaviors and female behaviors in our poison frog is developed through maternal imprinting. And so this likely explains why we're seeing this very concordant evolution between the behavior and the colomorphs in all Boccus-del-Toro populations. And so this also is, as I said, the first evidence ever that we know of that demonstrated imprinting in an amphibian, which I think is pretty exciting. And lastly, through the mathematical model, we have demonstrated that the combination of rival and sexual imprinting is a pretty effective mechanism that can create a scenario that is suitable for speciation to proceed. And so with these projects, I hope I have convinced you that both male-male competition and behavioral learning are important aspects of sexual selection in addition to female choice, and it's something that we should consider when we are thinking about the evolution of reproductive isolation or thinking about speciation. All right, so I will part with you with a couple of take-home messages. So first, I want to say that divergent sexual traits oftentimes mediates both male-male competition and female choice. And this is not something that is unique to our poison frog. This should be seen in a lot of animal systems. There are already some examples out there, but also I think a lot of our research efforts are focused on female choice. And if we actually look, we will see a lot more patterns that are associated with male-male competition. And this is an important thing because mating patterns is influenced not only by female preference, but determined by the interactions of female preference, male-male competition, and also likely the social context. So when we are thinking about the evolution of reproductive isolation, which is the ultimate result of mating patterns, we need to consider all of these things together. And lastly, through this last part of my talk, we have demonstrated that learned behaviors can have very different evolutionary outcomes compared to genetic behaviors. So we need to consider these, I guess, non-traditional ways of developing behaviors in speciation and on their role that contributes to speciation. Or more broadly, how does non-genetic behaviors, so things that are learned, things that are plastic, how that influence evolutionary processes. All right, so that concludes my dissertation talk, but before letting you ask questions, I have a lot of people to thank. So first and foremost, I want to thank the Richizawaki Lab. So here's our fearless leader, Dr. Corey Richizawaki. So I really need to thank Corey for welcoming me into her lab, and she has been a wonderful mentor in the past 6 years. So she has always urged me to be independent and creative in the research projects that I develop, but she also was not hesitant to lend a hand when I'm struggling or when I needed help. And so I just want to express my deepest gratitude to Corey. And I would also like to thank everyone else in the Richizawaki Lab, so all current and past graduate students, postdocs, lab managers, and undergraduates. You guys have been awesome in helping me a lot in my experiment, especially in managing the frog room, and also in giving me suggestions and comments on my projects, my presentations, and my manuscript, and also just being a great company in day-to-day work life. So thank you all. And next, I want to also thank all of my collaborators. So Heike and Simone, who I worked with back in my first field season. It was wonderful to work with you. And Matt Dugas, who helped me get my first field season going. And also we have collaborated a lot on developing projects. And so I think Dugas have guided me a lot in writing my first author research paper, and is instrumental to teaching me how to write clearly and effectively. So thanks, Dugas. And of course, Maria. So Maria have very generously hosted me in her lab for a month and taught me how to build a genetics model and how to analyze that. And so before that, I didn't have any training in building up any mathematical models. So I really need to thank Maria for introducing me to the world of mathematics. And she is also my external dissertation committee. So thanks, Maria. And I would also like to thank my dissertation committee here at Pittsburgh. So Wirt Carson, Mark Gervais, and Mark Turcott. So my dissertation committee have always urged me to put my research in a broader framework of ecology and evolution. So basically finding the big question. And that has always been a voice back in my head when I am starting a new project or started writing a new paper or putting together a presentation. And I think it's a really important training for PhD students. So thank you all for that. And of course, all the funding sources that make the projects possible. All right, so next I want to thank everyone that has helped me in the past four field seasons when I was in Panama, Bocas del Toro. So I want to thank Plinio, who is the scientific coordinator at the field station of Smithsonian Tropical Research Institute that helped with a lot of logistics of field work. And Sebastian, who is the contracted boat driver for Smithsonian, and also the local expert that knows where all the frogs are. So he actually took us to a lot of the field site that I worked in. And also, of course, all the undergraduates that were my research assistants in the field. So special shout out to Vincent, Mycia, Houston, and Sean, who did a lot of the heavy lifting of conducting experiments, and a lot of these would not be possible without them. And also just all of my friends that I made in BOCUS. So as you know, long field seasons can get lonely sometimes. And something as small as just getting a beer after a long day of field work is very significant to me. So thank you all. And next, I would like to thank my friends. So my support network here at Pittsburgh. So that includes the community in the biological sciences at University of Pittsburgh, especially the ecology and evolution community, and also my friends outside the department. So special thanks to Michelle, who was also a grad student that just graduated from the same department a couple of months ago. So Michelle has been a great friend, and also we have always urged each other to be more productive or just freak out together about grad school stuff. And it was wonderful to have you during my PhD, Michelle. And also special shout out to my friends that I play volleyball with. And so sometimes this is I think one of the most important things that kept me sane and happy and healthy in grad school. And of course, everyone else that cheered for my successes and helped me when I'm going through struggles during my PhD. All right, so last but not least, my family. So my parents and my younger brother. So unfortunately, they had to cancel their plans to travel to the US because of lockdowns. But the silver lining is, since I am defending remotely, they actually get to watch it back in Taiwan. And also Eric, who get to watch it in the West Coast, and he has to get up really early for that. And so my family is a bunch of fun, goofy, and interesting people. And I just want to say that I am really grateful that I am part of this family. And no matter where my career takes me, being part of this family will always be the anchor and the foundation of who I am. So I just want to say thank you for that. All right, so that concludes my talk. And I also want to thank you all for listening. And now I will gladly take any questions. Thank you. I hope the connection did not break out. Okay, yay.
All right, everything seems to have gone smoothly, so that's great. So
yay, okay, I was a little bit scared.
If you have a question for Yusan, please indicate it in the chat so that she can know who to call on.
Okay. Okay, so the question is about how assortative aggression might have evolved. So I think there are two levels, so the proximate level and the ultimate level. So the proximate level, so from what we have demonstrated, seems like from imprinting, because imprinting inherited the mating traits from the parents, and you also imprint on the mom. So to some degree, you will have this association between the coloration of themselves and their behavior. So that's, I think, the proximate mechanism of how that might evolve. And so the ultimate, I guess, mechanism will be harder to explain. So between different species, sorry, is somebody talking or? Okay, so in an ultimate mechanism, so if you think of it as between different species, these species, they will want to avoid aggressive behaviors with each other because it's not beneficial. But in a species that are going through speciation, it's more muddled because we're not sure whether the resource they're competing for is partitioned or not. So I think that's also something that could be investigated in the future. So basically, if aggression is allowed to evolve and female preferences are allowed to evolve, how might these two interact with each other, because they are related to the I guess the fitness of certain behaviors. Okay, I hope that answered your question.
Yeah, thanks. Just hi. Yeah, so hi, Maria.
Oh, it is this Justin. I was wondering
yeah,
what Justin that was.
Yeah.
I'm doing a lot of adjustments.
Yeah, so I was just wondering whether what you already have observed in your system might be sufficient to explain why it might have evolved. And it's not perfectly clear to me, but I was thinking that in some of my previous models with the evolution of imprinting on the parental phenotype, it actually evolves in the context of speciation because it helps avoid costly hybridization. So I'm just also kind of wondering whether something similar might have occurred in your system. But in your case, it's a negative frequency dependence instead of positive frequency dependence. So my intuition is not good enough to tell me whether it will work or not.
Yeah, that's what mathematical models are for, right?
Yeah.
Yeah, so I think it's a little bit different in a tiny bit different in our case because we don't have a clear evidence of any cost to being a hybrid other than in reproduction. So there's no survival costs. So evidence for survival costs so far in our system. And so, yeah, so in our model, it is a pretty simple model and the behaviors or the aggression strengths are not allowed to evolve. So I think if we allow that to evolve, we might see a lot of different patterns in different scenarios because there are fitness costs to having a higher preference, lower preference, and higher aggression and lower aggression, and also maybe the direction of that as well. So yeah, I hope that answered the question. So basically, there's a lot of like, I don't know, but it's fun.
Yeah.
Okay.
And great talk too.
Thank you. All right. Okay. Oh wait, I'm getting a little bit lost on the questions here. Let me just scroll down. The next question is from Calbert Desk on self-referent phenotype matching in a mixed color breeding experiment. So, so no, we did not specifically test for do you want to ask your question, elaborate more on your question, or should I just start explaining?
Sure. So this is Mark Hauber, and I was wondering if there was an interaction term between the mother's color preference versus the breeding treatment, because that would be consistent with some sort of genetic input and or with self-referent phenotype matching. But if there was no interaction term in your data, then there's no evidence for that.
Okay, so I don't think so. So in our cross-filtering experiment, so we have a pretty small sample size, so it's a little bit hard to tell apart on the specific patterns that are associated with certain color morphs. So that's one thing. And I think the other is, so we only test for, so in the mixed breeding treatment, we only test for preferences or aggression biases towards the mom's color and the dad's color, but we did not test for behaviors toward the mixed coloration. So that can manifest in a lot of ways depending on what color morphs we are mixing together. So I guess the question to this is also we don't have sufficient evidence to reject this hypothesis, but I think according to what we saw in the other two treatments, the most likely scenario, we still think it should be maternal imprinting instead of self-referent phenotype matching.
Great, thank you, great talk.
Okay. Thanks. Okay, so moving on to the next question from Danny Hughes, if I'm correctly pronouncing that. Do you want to elaborate on your question?
Yeah, hey, great talk.
Hi, thank you.
How are you doing?
Good.
So yeah, I was just curious if you measured anything else that may be like predictive of whether a male wins in a contest, like the brightness that it displays or the call intensity that it gives off, maybe some element that's associated with its overall fitness. Maybe that is predictive of whether it wins a battle.
Mm-hmm. Yeah. So great questions. I think a lot of these might be involved in the relative strength of mammal competition. So there are some research out there looking at in a monomorphic population, so a population with all the same color. So it seems like in those populations, the brighter you are as a male, you will lead to a higher aggression level and also a higher probability that they will win that stage contest. So that's one thing. So and body size. So body size, I do have another project that I didn't have the chance to present today that looked at how body size could have contributed to I guess male competition. And in that specific setup, I don't think I saw any evidence of body size contributing to male competition results, but also I did not specifically did that in my experiment in the second part, because I tried to make the conditions equal for two males, so we matched their size. Call intensity, I don't know, maybe. So these are a lot of things that I don't know, but potentially could contribute to those differences, I think. Sorry, there wasn't a very coherent.
I'm just curious if you measured these other elements. That's totally fine. Just curiosity. Nice
Yeah,
work.
so I think, another thing I think I can comment on is on the second experiment. So looking at the male aggression patterns that is across that transition zone. So instead of categorizing them by eye as like red, blue, and intermediate, we also use photographs to generate a color score and see if that influences their aggression. And so it seems like it's giving the same pattern as we have seen in categorical color variables. So we didn't particularly see brightness popping up as a determinant factor of aggression differences, I should say. Yeah.
Gotcha. Thank you.
Thanks. And next is Marco. Hi, Marco. Do you want to elaborate on your things?
Yeah, it's quite what I wrote to you, but I find amazing how the tadpoles can really learn about their father or mother color, right? So I was wondering if this imprinting, do you believe is really costly in cognitive related in their brains, I mean? And at some point you talked about that the tadpoles are, at some point the father is taking care of them, and at some point is the mother. So if the stage of the tadpole has something to do or anything like that.
Yeah, great question. So this is something that I wondered a lot about as well. So I think I'll first comment on the benefits and costs of imprinting. So what I can think of is because the tadpoles are in a field, they're not only encountering their mothers, there are also other things that visits those water bodies, and potentially those could be predators of the tadpoles. So I think intuitively, although I don't know if this is true, these tadpoles should respond more to their mother and freeze when there's a predator nearby. So that might be the benefit of imprinting on the mom's color and responding more, and that might carry over to adult stage. So this was just a hypothesis. I don't have any evidence to back this up. So that's just one of the things that I thought might be possible. And so the other thing is, so in our poison frogs, the father takes care of the eggs from before hatching to very early in hatching. So the other related thing is whether the brain of the tadpoles or the visual system of the tadpoles is developed enough at that stage to have any kind of imprinting. So it would be really interesting to compare this with other poison frog species, for example, that has paternal care more than maternal care. So because some of them, they do, the primary caregiver, I guess, will be the dad, obviously not through unfertilized eggs, but will be taking them to water bodies and sometimes guarding them. So I think it'll be really interesting to explore the differences in these related species, but with different parental care and mating systems. Does that answer your question?
Yeah, that's great. Thank you.
Thank you. Okay, hi, Jess.
Hi, Yusan. Amazing talk. Thank you so much. That was really, really inspiring.
Thanks.
Did you get my question? So I asked whether you would say that the aggressive loser is showing spite. And was there anything that you measured that predicted whether or not the loser became aggressive or submissive? Because that was such a cool observation. And I just wanted to hear more about what you thought was going on there.
Yeah, that was that kind of like the unexpected result that we got from the experiment in a very controlled setting. But I think it's really cool as well. So the, I guess you could say, goal of them doing it out of spite or other things, or they were just like trying to get any matings that is potentially possible. But that is also something that we saw sometimes in the field. So maybe manifesting as a sneaker male in a male's territory. So in the field, in this species, what have been observed is that there are sneaker behaviors, but there are no, I guess, documented cases of these behaviors leading to successful matings. So yeah, I don't know whether that's just because it's very rare and it's hard to observe, but based on our control breeding experiments, it's really surprising that these behaviors, assume that it is costly, they don't actually result in a higher reproductive success. So I don't actually know why they did that.
Well, maybe I wonder whether if because your tanks were quite small, like maybe in a natural territory, maybe it would have resulted in mating success. But
maybe. And also, I think there's the point that in our tank setup, the losers are not allowed to leave the territory, but in the field, these losers might be able to leave and find another territory that they could potentially take over, or they could maybe, I don't know, easily to sneak matings in. So yeah.
Very cool. Very cool. Thank you.
Thank you. And hi, Rainey. And hi, Michelle.
Hi. So again, like amazing, wonderful talk, unsurprising, but well done. I guess I just wanted to hear a little bit more about in what scenarios you could predict, since now we know that like amphibians have imprinting too. Do you think there are any particular traits or behaviors that might be like increase the chances that you're going to see imprinting in other species, and like where might you look next to see other imprinting patterns?
Yeah, great questions. So, oh boy, this is a big question. So I think, so for imprinting to happen, you have to have, I guess, parental care would be a good predictor of where parental imprinting is happening, and depending on like the identity of the individual taking care of the offspring, so mom or dad, that could change on the imprinting pattern. So like in our frogs, the mom is disproportionately actually taking care of the tadpoles more than that from the dad. So that is perhaps why we see maternal imprinting and not paternal imprinting in our systems. So in other systems that the dad takes care of the offspring, then that could lead to paternal imprinting. And I think even for species that doesn't have parental care, it is also worth investigating whether the experiences during a tadpole stage, whatever that might be, so maybe like coexisting individuals in the same area or other things, or other olfactory cues, or I don't know, acoustic cues. I don't know, I'm just like talking gibberish here, but I think it's worth investigating how learning, even in situations where we don't normally think of, could have influence behaviors when these tadpoles go into adults, or maybe just like more generally when during larvae stage and adult stage.
Yeah. Oh, that's so cool. Because it could even be like not visual in some other species.
Yeah, definitely.
That's awesome. Thank you.
Thanks. Okay. Justin again. I think is this the same Justin?
Yeah.
Hi.
Hi. Yeah, I was just kind of following up from an earlier question about spite, because I know there are some models out there showing that spite could have evolved through some inclusive fitness models. So I'm wondering how far do they disperse, and whether they have to compete with their kin, or maybe they're more likely to target unrelated individuals, for example.
Huh. Okay. Cool point. So I think what I can comment on is so far the evidence in this species showed that the individual seems to not be able to tell apart different individuals. So that's through the deer enemy effect kind of experiment. So looking at whether males distinguish between familiar individuals and unfamiliar individuals. And seems like at least in the population being investigated, that wasn't something that is shown. But yeah, so I think that's the evidence I can think of that's related. But about dispersal, I think so amphibians generally don't disperse very far from their original place. And also males tend to stay in the same area in their territories, and females tend to have a home range as well, which is a little bit larger and overlaps several of the males. So there is, I think, on a paper that should come out at some point, looking at the genetic line in that polymorphic region. Is that right, Corey? Yeah, and so that should tell us something about the I guess the dispersal distance or the genetic patterns in those areas.
Cool. Thanks.
Okay, thanks. Okay, does anyone else have any other questions?
Yusan, I see just one left that I think we
Oh,
might
okay.
have skipped over from.
Oh, sorry. Did I skip over someone?
Yeah, there's one that said, also was there a viability loss for the mixed color breeding tadpoles compared to the pure color pairs offspring?
Where is that? I seem to miss that.
It's up at the beginning.
Okay. Oh, okay.
10:05.
Oh, yeah. No, so there's also actually a paper out there also from Corey's lab looking at the viability and the reproductive fitness of these mixed color breeding pairs, and seems like there isn't any cost to being a colomorph that is from two different colomorphs, and also they don't suffer from reduced fertility as well. So it seems like there isn't any, at least there isn't any evidence in captive breeding about that is related to mating between the different colomorphs. Okay.
Right. I think that
Thanks,
is
Sarah and Swan.
all of the questions. So at this point, we'll thank everyone for coming and wish that we could have all seen you in person, but thank you for taking time out of your day to hear Yusan's dissertation talk. If you are not a member of the committee, we'll ask you to leave the meeting at this time.